So, today we're going to discuss about the hypersensitivity. In the previous chapter, the immune response has been described as a defense mechanism by which the body rids itself of potentially harmful antigens. In some cases, however, this process can end up causing damage to the host. When this type of reaction occurs, it is termed hypersensitivity, which can be defined as a heightened state of immune responsiveness. Typically, it is an exaggerated response to a harmless antigen that results in injury to the tissues, the diseases, or even death. British immunologists PGH Gel and RRA Combs devised a classification system for such reaction based on 4 different categories. In type 1 reactions, cell-bound antibody reacts with antigen to release physiologically active substance. Type 2 reactions are those in which free antibody reacts with antigen associated with cell surface. In type 3 hypersensitivity, antibody reacts with soluble antigen to form complexes that precipitates in the tissues. In these latter two types, complement play a major role in producing tissue damage. Type 4 hypersensitivity differs from the other three because sensitized T cells rather than antibody are responsible for the symptoms that develop. Although some disease manifestation may overlap among these categories, knowledge of the general characteristics of each will help in understanding the immune process that triggers such tissue damage. Type 1 through Type 3 have previously been referred to as immediate hypersensitivity because symptoms develop within a few minutes to a few hours. Type 4 hypersensitivity has been called delayed hypersensitivity because its manifestations are not seen until 24 to 48 hours after contact with antigen. For each of the 4 main reaction types, the nature of the immune reactant is discussed. First, we must discuss the type 1 hypersensitivity. The distinguishing feature of type 1 hypersensitivity is the short time lag, usually seconds to minutes, between exposures to antigen and the onset of clinical symptoms. The key reactant present in type 1 or immediate sensitivity reaction is IgE. Antigen that triggers formation of IgE are called atopic antigen or allergens. Atopy refers to an inherited tendency to respond to naturally occurring inhaled and ingested allergens with continued production of IgE. The prevalence of allergic disease has increased greatly in developed countries in the last 20 to 30 years, and it is estimated that as much as 30% of the US population have some type of allergic symptoms. Typically, patient who exhibits allergic or immediate hypersensitivity reaction produce a large amount of IgE in response to a small concentration of antigen. IgE levels appear to depend on the interaction of both genetic and environmental factors. Carl Wilhelm Protsnetz and Heinz Kostner were the first researchers to show that a serum factor was responsible for a type 1 reaction. Serum from Kostner, who was allergic to fish, was injected into Protsnetz. A later exposure to fish antigen at the same site resulted in an allergic skin reaction. This type of reaction is known as passive cutaneous anaphylaxis. It occurs when serum is transferred from an allergic individual to a non-allergic individual, and then the second individual is challenged with specific antigen. Although this experiment was conducted in 1921, and it was not until 1967 that the serum factor responsible, namely IgE, was identified. Characteristics of IgE are described in Chapter 4. Now we're going to discuss about the triggering of type 1 reaction by IgE. IgE is primarily synthesized in the lymphoid tissue of the respiratory and gastrointestinal tracts. Normal levels are in the range of approximately 150 ng per ml. The regulation of IgE production appears to be a function of a subset of B cells called type 2 helper cells, or Th2 in acronym. The normal immune response to microorganisms and with possible allergens is a function of type 1 helper cells, or Th1, which produce interferons gamma, IFNY, interferons gamma along with interleukins 12 and interleukins 18, which are produced by macrophage, may actually suppress production of IgE type antibodies. However, in people with allergies, Th2 cell respond instead and produce interleukins 3, interleukins 4, interleukins 5, interleukins 9, and interleukins 13. See Chapter 5 for a discussion of cytokines. Interleukins 4 and interleukins 13 are responsible for the final differentiation that occurs in B cells, initiating the transcription of the gene that codes for the epsilon heavy chain of immunoglobulin molecules belonging to the IgE class. Interleukins 5 and interleukins 9 are involved in the development of eosinophil, while interleukins 4 and interleukins 9 promote development of mast cells. Interleukins 4, 9, and 13 all act to stimulate overproduction of mucus, a characteristic of most allergic reaction. This propensity to secrete cytokines that promotes production of IgE is linked to a gene locus on chromosomes 5 that encodes cytokines, interleukins 3, 4, 5, 9, 13, and granulocytes monocytes colony stimulating factor, or GM-CSF. All of these cytokines are key to a switch to a T helper cells to response. Interleukins 4 and 13 activates transcription of the epsilon gene in B cells when they bind to specific receptor. So here are the comparisons of hypersensitivity reaction. We have immune mediator, the IgE is the type 1, IgG is the type 2, type 3 is IgG or IgM, and type 4 is T cells. In the antigen, we have that the type 1 of antigen is heterologous, and the type 2 is autologous or heterologous, and the type 3 is autologous and heterologous or It's the same thing to type 3 and 4 on the type 2. In the complement involvement, in type 1 is no, type 2 is yes, type 3 is yes, and type 4 is no. In the immune mechanism, type 1 is that release of mediator from mast cells and basophil, while the type 2 is cytolysis due to antibody and complement, and type 3 is that the deposit of antigen-antibody complexes, and the type 4 is the release of cytokines. An example for this is that the anaphylaxis have fever, food allergies, asthma, while the transfusion reaction you know what I mean. Although actual antibody synthesis is regulated by the actions of cytokines, the tendency to respond to specific allergens appears to be linked to inheritance of certain major histocompatibility complex, or MHC genes. Of various human leukocyte antigen or HLA class 2 molecules, especially HLA, DR2, DR4, and DR7, seems to be associated with a high response to individual allergens. HLA-D molecules are known to play a role in antigen presentation, and thus individuals who possess particular HLA molecules are more likely to respond to certain allergens. In addition, Individuals who are prone to allergen exhibit certain variation in the gene found on chromosome 11q that codes for receptor for IgE, found on several types of cells. It is thought that polymorphism in the beta chain of this receptor are linked to autophagy. This high affinity receptor named FcER1 receptor bind the fragment crystallizable Fc region of the epsilon heavy chains and are found on basophil and mast cells. A single cell may have as many as 200,000 such receptors. Other cells such as monocyte eosinophil, Langerhans cells, and dendritic cells also have receptors for IgE, but their concentration is low. However, Langerhans and dendritic cells internalize and process allergens from the environment and transport the allergens MHC class II complex to local lymphoid tissue, where synthesis of IgE occurs. Binding of IgE to cell membrane increases the half-life of IgE from 2 or 3 days up to 10 days. Once bound, IgE serves as an antigen receptor on mast cell and basophil. Cross-link of at least 2 antibody molecules by antigen triggers release of mediator from these cells. When cross-linking occurs, intracellular signaling events due to multiple phosphorylation reactions cause an increase in calcium. The influx of calcium promotes synthesis of arachidonic acid from membrane lipids. It also promotes cytokine synthesis and release of preformed mediators due to degranulation. Now we're going to discuss the role of mast cell and basophil. Mast cells are the principal effector cell of immediate hypersensitivity, and they are derived from precursors in the bone marrow that migrate to specific tissue site to mature. Although they are found throughout the body, they are most prominent in connective tissue, the skin, the upper and lower respiratory tract, and the gastrointestinal tract. In most organs, mast cells tends to be concentrated around the small blood vessel, the lymphatics, the nerves, and the glandular tissue. These cells contain numerous cytoplasmic granules that are enclosed by a bilayer membrane. Histamine, which comprises approximately 10% of the total weight of granular constituent, is found in 10 times greater supply per cell than in basophil. Mast cell population in different sites differ in the amount of allergic mediator that they contain in their sensitivity to activating stimuli and cytokines. All types of cells are triggered in the same manner, however, they release a variety of cytokines and other mediators that enhance the allergic response. Basophil represent approximately 1% of the white blood cell and peripheral blood. They have a half-life of about 3 days. They contain histamine-rich granules and high-affinity receptor for IgE. Just as in mast cells, they respond to chemotactic stimulation and tend to accumulate in the tissues during an inflammatory reaction. In this presence of IgE, the number of receptors has been found to increase, indicating a possible mechanism of upregulation during an allergic reaction. Now we're going to discuss the mediator release from granules. We have first to discuss the performed mediators, which is the cross-linking of surface-bound IgE on basophil and mast cells by a specific allergen cause changes in the cell membrane that results in the release of mediator from the cytoplasmic granules. These performed or primary mediator include histamine, heparin, eosinophil, chemotactic, factors of anaphylaxis or ECFA, neutrophil chemotactic factor, and protease. Release of this substance is responsible for the early phase symptoms seen in allergic reaction, which occur within 30 to 60 minutes after exposure to the allergens. The effect of each of these mediators is discussed in a summary